Dear students, welcome to the e-learning sessions of semester 3rd, subject Pharmacognosy and Phytochemistry 1. In previous lecture 1, we have already covered about the marine pharmacognosy, and in this lecture 2 of semester 3rd, I will cover the topic plant products, and in plant products I will cover about the fibers, which includes cotton, jute and hemp. Now, what is the context of this chapter and what are the points you should study under this? Are the study of the biological source, chemical nature, uses of the drugs of natural origin, that is the plant product of these following drug. This drug means plant products in fibers, cotton, jute and hemp. So, in short, we'll study about the biological source, chemical nature and uses of drugs of natural origin like cotton, jute and hemp. Now, what are the contents we'll study in this topic or under this topic? First one is the introduction, which includes what are plant fibers, how they are developed, what are the differentiation of fibers like phloem fibers, xylem fibers, we'll discuss about that. Then the classification of fiber. There are different fibers like from animal origin, from plant origin, and we will study in this plant origin. Identification of fibers. How will we identify the fibers? By the help of different tests, physical, chemical, microscopical. Under this we will study all this. Now the biological source, chemical nature and uses of all these three, that is cotton, jute and hemp. Now first, introduction. What are fibers? Fibers in pharmacognosy is defined as the elongated, means long, thick walled cells with pointed ends. So fibers are the elongated, thick wall cell with pointed end, and this cell wall consists of cellulose. So the major constituent present in the fiber is the cellulose, and this cellulose may or may not contain lignin. What is the main use of the fiber? They are generally used for the surgical dressings made up of natural or the artificial material. Now, introduction. Fibers obtained from many sources. They can be obtained from for example, we are studying the plant fibers. So they are obtained from plant. Example of jute, flax, banana, cotton and hemp. From all these five, we will study jute, cotton and hemp as per your syllabus. So we are studying only the plant fibers. But for your knowledge, you should know that the fibers are of 4 types: plant fibers, animal fibers, mineral fibers and synthetic fibers. So first, the elaboration will do about the plant fibers. And you should know the example of different types of fibers. How the fibers are developed? Fibers are developed from a single cell which grows rapidly in the axial direction, straight the way direction. During this growth period, the tips are elongating cells. Fibers are the elongated, so their tips are the elongating cell and they may push one another. Other fiber is push the another fiber, and the fibers are unicellular in structure. They are the single cell structures. Now, how will we differentiate the fibers? Fibers are basically differentiated into the 3 types. One is pericyclic fibers. So the fiber presents in the pericyclic region are known as pericyclic fiber. Example, flax and hemp are the example of pericyclic fibers. Next is the phloem fibers. Fiber present in the phloem region is known as the phloem fibers. Example is jute. And xylem fibers are also called as wood fibers. They are the fibers present in the xylem region of plant. Therefore, they are called as xylem fibers. So here we have studied three types of fibers: phloem fibers, pericyclic fibers, and xylem fibers. Now, the classifications of fibers. We should be clear that this is the whole classification of fibers: natural fibers and artificial fibers. And natural fibers include from the natural sources: plant, animal, and the mineral. And in plant, for example, cotton, jute, hemp, flax, and banana. And in our syllabus, we'll cover the cotton, jute, and hemp from plant sources. Next is the animal source. So, animal source from wool and silk. And another is the mineral. Glass and asbestos are the example of mineral fibers. So, in natural category, there are three categories: plant, animal, and mineral. But we will study only about the plant. Now next is artificial. Artificial means created fibers. They can be regenerated or synthetic. The regenerated fibers are of two types: carbohydrate and protein. And in carbohydrate category, there is alginate, viscose, proxylene, acetate rayon. And in protein category, fibrillin type. And in artificial, regenerated is in carbohydrate and protein, and synthetic is in nylon and terylene. So basically you all should be clear that fibers are of two types: natural and artificial. Forget about artificial. Here we are emphasis on the natural, and in natural also we are emphasis on the plant fibers. Got the point? Next, identification. How will we identify the fibers that this contain the fibers? So they can be identified by the different analytical procedures. First one, the most common, is the microscopic examination. By the help of compound or different SEM, means scanning electron microscope, we can observe the fibers, their elongated cell, their thick walls that contain the cellulose. By all these parameters, we can observe them. Physical test, by performing the physical test like ignition test. Another is chemical test, by solubility test. Depends on the solubility, we can confirm that it is the fiber. Or with the test with the reagents, means test with the chemicals, we can also prove that the substance contain the fibers. Next, the first category of plant fibers, that is natural plant fiber, is cotton. The synonym for cotton, as you all know, it can called as raw cotton, cotton wool, purified cotton, absorbent cotton, because cotton absorbed, surgical cottons. These are the different types or the name by which the cotton can be called as. This, you all can see the cotton plant photograph here. What is the biological source? Cotton consists of the epidermal trichomes or the hair of the seeds of cultivated species of Gossypium herbaceum, and family is Malvaceae. So you should remember the synonym: biological source. And then geographical source. It is produced commercially in USA, Egypt, and in India, and it is also cultivated in various parts of Africa and South America. So the major portion in this is, you should know the synonym: biological source. Okay. Next, how should we prepare the absorbent cotton? So cotton is mainly and commonly grown for the purpose of fibers. So each capsule contains numerous seeds, and these seeds are covered with the hairs called as balls, and these balls are collected, dried, and taken to the ginning press. In this press, trichomes are separated from the seeds. So for preparing the absorbent cotton, we have separated the trichome from the seeds. Now various devices nowadays, analytical devices and various controlling devices are there to separate the hairs of the cotton. The short and long hair are separated from each other. The hair with short length are called as linters, and these linters are used for manufacturing of absorbent cotton, and the long hair is used for the preparation of the cloth. The raw cotton obtained, which contain the impurities like fat, wax, vegetable debris. So every portion and each portion has certain advantages Now the preparation of absorbent cotton, it is processed to get rid of the most of the impurities. We can process so that the impurities can be removed out. It is taken to the machine known as cotton opener. There it is treated with dilute soda as solution under the pressure of 10 to 15 hours. The wax, fatty material and the coloring materials, these all are called as impurities, are removed by the treatment. Then it is washed with water, treated with the bleaching agents, then again washed with water, dried and compressed in the flat sheets. It is finally sterilized. The main important point, it should be sterilized, packed in the wrappers. By the means of radiation, we can sterilize the cotton. So this is the complete procedure for the preparation of the absorbent cotton. What are the morphological characteristics of the organic or the organoleptic properties of the cotton? Color is white due to bleaching. Otherwise, the cotton which we obtain from plant is little yellowish color, but due to bleaching we can convert it into white. Order is orderless, tasteless. Size is 2.5 to 4.5 centimeter in length and in micron 25 to 35 micron in diameter. It may be slightly off-white in color if sterilized. Now standard parameters for the absorbent is water soluble extract should not be more than 0.5%, sulfated edge should not be more than 0.5%. It should comply the fluorescence test, acidity and oxidizing substance. The most important part is the chemical constituent. In the cotton, 90% part is of cellulose, 7 to 8% is the moisture. So how to perform the chemical test of the cotton? There are many methods for performing the chemical test, but the soak cotton fiber simple method is dipped in iodine water and dry and add few drops of 80% sulfuric acid. Sulfuric acid means H2SO4. This trichomes, if cotton is there, the trichome show purple or bluish green color. This shows it's distinguished from the other fiber. If this color shown, then it means it is the cotton. And cotton is insoluble in dilute sodium hydroxide, means NaOH solution, and HCl. It can be distinguished in this way by this silk. Now, what is the use of cotton? It is used as a filtering medium and in surgical dressing. It is also used as an insulating material. And absorbent cotton, as the name suggests, absorb the blood, so it is used in dressing. It can absorb blood, mucus, pus and prevents the wound from the infection. Next is, as I have told you, cotton is the 90% cellulose. So this is the structure of cellulose chain. You all should remember and can also draw the structure. This is the OH group, this is the H group, and this is the CH2 OH point, and this is the chain of the cellulose. You can draw only one structure also to showing the cellulose structure for the cotton. Now the second one is the in the plant products and natural categories, the jute. The biological source for the jute is, I have already told you that the differentiation of fiber is of three types: pericyclic, xylem fiber and phloem fiber. So the jute, in phloem fiber I have told you the example is jute. So here it comes that it consists of the phloem fibers of the stem of various species of the Corchorus olitorius or Corchorus capsularis. The family is Tiliaceae, and the geographical sources, basically the jute is cultivated in West Bengal, and morphology is, color is yellowish brown, it is tasteless, odorless, and this is the preparation method of the jute. Retting is the name of the process for the preparation of the bast fibers. This process is done by three methods, that is microbial, steam or by the mechanical process. And the microscopy of the jute is, TS of the strand when treated with HCl, fluoroglucinol, strands the deep red color. This deep red color shows the presence of the lignin, and which is surrounded by the lumen with various sizes. These strands can be separated by treating it with the mixture of KCl. KCl means potassium chloride and nitric acid. So this is the microscopy by which TS can be checked. Chemical constituent. Jute fibers are composed of the plant material cellulose and lignin, that is 50 to 53% of the cellulose, nearly 20% of the hemicellulose, 10 to 12% of the lignin, and little amount of moisture, fat, wax and ash includes the impurities, and the major portion is 50 to 53% of the cellulose. The main use of jute is, it is the second most important vegetable fiber after the cotton, and this jute is used in the manufacture of the filtering and straining medium, padding splints, and is also used for the preparation of the coarse bags. Jute is the most common. Next, the natural category in plant product is the hemp. The geographical source of hemp is the raw material imported from China, America, Germany, Switzerland, and the biological source of hemp is the pericyclic fiber. So in pericyclic fiber, when you will see, you can see the example there is hemp. So you can correlate there the differentiation of fiber and the name of the fiber. So its source is Cannabis sativa. The main constituent obtained in hemp is cellulose and lignin. And the use of hemp is has a variety of uses, mainly in its tensile strength is good, so it is ideal for the use of the tea bags, industrial filters for our pharmaceutical use. It is used in the industrial filters or in microbiological, sometimes filter as a filter. Hemp is used as a filter, bast fibers, and this completes your all the three drugs: cotton, jute and hemp. Now comes to the question, what are the important questions which may be asked from this? So sometimes maybe in exam they can ask about the short note on plant fibers. So you can write all these, the definition of plant fiber, in shortly the classification of fiber, that is natural and plant fibers, and in that you can explain the cotton, jute and hemp in a short form, only the chemical constituent and the uses, name, biological source, chemical constituent and use. This much you can explain in the short note. And define fibers, classify them. So you can write the fibers in detail and then classify that tabular or that representation of the classification. Then the question can also come like that, write a note on cotton and hemp. So you can also write, I have already given you the details of the cotton and hemp. Explain different types of the plant fiber. So you know what are the different types of plant fiber. They are natural and artificial plant fibers. So you can explain that also. These are the difference from where I have taken the matter. So you can go through that, and if in case of any queries, you can ask at any point of time. And one more thing is relating to notes or PDF. I have everything matter and every matter in a arranged form. So I will give you the notes, more elaborated notes than I have presented, because here I have presented in slides, so I can't fill the slides with the matter. So I have explained you more and given the slides in a short form, but if you want more matter, I can give you later. Okay, thank you once again. Be safe and stay healthy. So please subscribe our YouTube channel Pharma Ignite and watch the lecture for the coming e-series. Thank you. Bye bye.